Greetings everyone and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Modeling Lipid and Glucose Metabolism Using Human iPSC-derived Hepatocytes. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The recapitulation of native cellular and physiological behavior is critical for accurate phenotypic screening and target identification, techniques which are paramount to both academic and biopharmaceutical researchers. Within the study of metabolic diseases, limitations of classical in vitro cell models are becoming increasingly evident, causing investigators to seek out improved alternatives. Induced pluripotent stem cells, or iPSCs, have provided investigators with reproducible, well-characterized, and a readily available source of various human cell types that are the logical alternative to current human cell models, thus ensuring advancements in basic biology and applied biomedical research. Let's meet our panel for this GEN webinar, who will illustrate how human iPSC-derived hepatocytes can facilitate research into human metabolic disorders. Our first speaker today is Kobe Carlson, Strategic Marketing Manager at Cellular Dynamics International. Dr. Carlson will provide us with background information on iPSC-derived hepatocytes and the numerous applications where these cells can be and are currently being utilized. Our second presenter is Siobhan Mullaney, Director of Translational Biology at Sanford Burnham Prebys Medical Discovery Institute. Dr. Mullaney will discuss her work using iPSC-derived hepatocytes in the study of non-alcoholic fatty liver disease. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all the particulars out of the way, let's get our presentation started. Kobe, take it away.
All right, thank you, Jeff, and thanks to everyone for joining us today. I would like to start by introducing you to the liver. The liver is a vital organ that supports nearly every other organ in the body in some way or another. It is estimated that there are nearly 500 different functions of the liver, and several of them are listed here. These functions range from controlling the levels of fats and amino acids and cholesterol and glucose in the blood to storing vitamins and minerals and other essential chemicals. It also produces bile and breaks down food and converts it into energy, and while at the same time neutralizing and destroying drugs, toxins, and infectious particles. Hepatocytes are the most abundant cell type in the liver, and they are responsible for performing the majority of these many different functions. Because of the importance of the liver and the role that hepatocytes play in human physiology, it should not be surprising that a great number of people are interested in research using this cell type. In fact, if you are registered for this webinar today, it is highly likely that you have used or are planning to use some type of hepatocyte-like cell pictured here. And examples of existing hepatocyte cell models are transformed cell lines like HepG2 or the HepaRG cell line, primary liver cells from an animal like a rat, and of course, the gold standard primary human hepatocytes, often abbreviated here as PHH. So these different hepatocyte-like models are being used in all facets of biomedical research, including bioengineering, cell therapy and regenerative medicine, infectious disease, specifically hepatitis, and safety toxicology studies. And a relative newcomer to this field are hepatocytes derived from human iPSCs, or induced pluripotent stem cells, which are going to be the topic of this webinar today. Okay, so iPSC technology has been around for over a decade now already, and many reviews are in the literature, and there are simple overviews summarizing this technology all over the internet. So therefore, I will keep my description fairly brief. And while there have been numerous improvements in the methodologies along the way, the basic principle has pretty much stayed constant and is illustrated here in this schematic. The starting point is an adult cell, preferably a blood or skin sample, that can be obtained from any individual, and that is any human donor. And these cells can be exposed to known reprogramming factors to induce them to revert back in time, basically back to a pluripotent stem cell state. And at this point, iPS cells can propagate indefinitely as well as be manipulated genetically with tools such as TALENs or CRISPRs. And then through various differentiation protocols for cell specialization, these stem cells can be instructed to give rise to just about every other cell type in the human body. And it is these methods, these core competencies needed to generate and maintain and genome engineer and differentiate iPS lines that our company, CDI, headquartered here in Madison, Wisconsin, was founded upon. And it is this transformative potential of iPSC technology, essentially how it is enabling for drug discovery, safety toxicology, and regenerative medicine. That was the reason, really, that Fujifilm acquired CDI over a year and a half ago, nearly 2 years now. Okay, next, where might iPSC-derived cells contribute to the success of your specific project or fit into your current workflow? Well, if you're using one of these commonly employed hepatocyte cell models introduced a couple slides ago, you are obviously familiar with the benefits and advantages of primary human hepatocytes, or HepG2s even, for example. But you are also probably aware of the limitations. So this slide here addresses some of the feature benefits of iPSC-derived cells in comparison to the more traditional options. And you can see here in the red text boxes that the limited availability and high variability of primary human cells, the lack of key functionalities of recombinant or transformed cell lines, and the poor representation of human diversity or the translation to the human condition with animal models has really underscored the importance of iPSC technology and has motivated researchers to consider other sources. So why iPSC? Well, simply put, one of the clear advantages of iPSC technology is that it offers the unique possibility of an unlimited and renewable source of hepatocytes from theoretically any individual donor. In addition to access to such supply and diversity, the fact that iPS cells can give rise to virtually any cell type in the body, I think will prove quite valuable, and we will touch on this later. But since it is really probably unreasonable to expect hepatocytes, either iPSC-derived or primaries, to gain mature functions or to retain their existing functionality in isolated monocultures grown on a flat 2D surface, it's really more likely that co-culture or 3D scaffolds or bioengineered devices will be required to build a better in vitro liver model. And it makes really logistical sense to leverage the same donor iPSC line to make other cell types necessary to populate such complex systems down the road. And finally, one of the true promises of iPSC technology is this concept of modeling human disease in a dish and using iPSC-derived cells to not only understand mechanism of the pathophysiology, but also to identify a measurable disease phenotype and perform library screening against that phenotype in an assay. And one way to really see this idea through to a reality is to ensure consistency. And I was reminded recently of this phrase, consistency is born by process. And I think that's where CDI comes in. And we're all about process here at CDI. And the industrialized protocols that we have developed here allow for the production of human hepatocytes at a quantity, quality, and purity that is really unmatched in the field. And, I think admittedly, hepatocyte differentiation and subsequent cryopreservation of the cells is very challenging. And the level of functionality expected has a bar that has really been set quite high by mature adult primary human hepatocytes. So while the integration of stem cells into labs using hepatocytes or hepatocyte-like cells has been described as promising or a work in progress, we think there have been some exciting developments in terms of longer survival in culture and improved function that we'll share about in the next few slides. And although I only focus on a few of these features really centered around glucose metabolism, I think that these characteristics support the story that Siobhan is going to present in the second half of this webinar today. Okay, so CDI has presented on the characterization of our hepatocyte product before, and they are known as iCell hepatocytes. So I am intentionally going to be a bit light here because this extensive data set can be viewed elsewhere, and that's not really the intent of this webinar. But suffice it to say that these cells express all the traditional markers such as HNF4 alpha and AAT. They make and secrete both albumin and urea over time. They express phase 1 metabolizing enzymes like CYP1A2, 2C9, 3A4, and I don't have this P450 data on this particular slide, but if you look over here on the left-hand side, the iPSC-derived hepatocytes really have a beautiful cell morphology where they exhibit the typical cubic or cobblestone shape. There are tight junctions and extensive bile canaliculi formation, as indicated by the yellow arrows, and there's clear binucleation. You can see the cells circled in blue. So importantly, we use all of these different characteristics of the cells to generate release criteria. And we have a QA/QC department here at CDI that evaluates every batch or lot of cells that we manufacture. And I think that really the results are pretty good because by looking at the picture that's below on the left, that are primary human hepatocytes in culture, those two images, they look very comparable. Maybe perhaps not even unreasonable to suggest indistinguishable. But like I said, reproducing all or even most of the functions of the gold standard primary human hepatocyte is a major hurdle that none of the current protocols out there have achieved. And I'm really including our own here. But I'd just like to go through a few more examples of how the implementation of iPSC-derived hepatocytes has advanced research and has overcome some of the problems associated with other models. First, there are some clear wins with respect to long-term viability in culture with these cells. In 2D, there's no need for matrigel overlays or collagen sandwich cultures, and they regularly last at least 2 weeks in culture. And I'm not showing that data right here, but what you're seeing now is if we make 3D spheroids using a protocol developed recently, we can easily keep the cells out to 4 or 5 weeks as pictured here. And this makes them suitable for long-term toxicity studies with repeat dosing, for example. Next, we have high confidence in our lot-to-lot consistency, and many of the assay protocols that we are developing to work with these cells are robust across multiple different donors. As mentioned earlier, making different cell types from a single iPSC line and using them to build a more complex co-culture system is in the works. And pictured here are iCell hepatocytes together with iCell macrophages, which exhibit phagocytic properties and are thought to operate like Kupffer cells. These data here compare the basal P450 enzyme activity of primary human hepatocytes as determined by LCMS to that of iPSC-derived hepatocytes. 2 takeaways here. One, the different samples of primaries display significant variation. And you can see the black circles here. There's a nearly 10-fold distribution in the data. Whereas, second takeaway, the improvements that we are making to hepatocyte culture, including media optimizations and 3D spheroid culture, are finally putting the CYP activity of these cells within an acceptable and comparable range, And this data is really quite tight. You can see the green triangles here. Finally, hepatotoxicity has been one of the primary applications for our hepatocytes. And we have contributed to some very nice studies as of late, like the one here shown in collaboration with molecular devices. But the possibilities with these cells goes way beyond tox. And this brings me to the next slide. We just talked about long-term hepatotoxicity, which is really being enabled to a great extent by 3D spheroid cell culture. And so it's appropriate that those two images go side by side. These hepatocytes can also be transduced using tools like BACMAM to express GFP in this example. And they can also be transfected with siRNA oligos against GFP here to modulate gene expression. We have placed these cells on various instruments and developed protocols to get you started testing them. And that includes the Seahorse XF analyzer to measure the bioenergetic properties of iCell hepatocytes. Moving on, we have also used HTS-compatible assay technologies such as HTRF or alpha-LISA to interrogate cell signaling pathways and quantify certain biomarkers such as PCSK9, pictured here, and that is released following treatment with atorvastatin. We showed on the previous slide basal P450 activity, but CYP induction is also improving in these cells. And lastly, human iPSC-derived hepatocytes are proving to be a unique and powerful system to model the infectivity of hepatitis viruses like HBV and HCV. Okay, now that we have covered all that data to hopefully put us on the same page here for the remainder of the webinar, we are ready to get into hepatic glucose metabolism. And basically what this schematic shows us is that the hormones insulin and glucagon work together by acting on the liver to maintain stable blood glucose levels. And when your blood sugar is too high, the pancreas releases insulin, which signals to the hepatocytes in the liver to stop making glucose by either storing more of it away as glycogen or by downregulating the metabolic pathways that make it, such as glycogenolysis or gluconeogenesis. Glucagon, on the other hand, has the opposite effect to that of insulin and can stimulate an increase in the concentration of glucose in the bloodstream when it is too low. Now, because of the many liver functions, such as the breakdown of proteins and lipids, there are many non-carbohydrate precursors around in this organ and available as gluconeogenic substrates, including alanine and glutamine, which are amino acids, glycerol from triglycerides, and other metabolic byproducts like lactate and pyruvate. So we figured that if we wanted these iPSC-derived hepatocytes to recapitulate one of the more important functions of a liver cell, we should really develop an assay for gluconeogenesis. And that is exactly what we did here. So let me walk you through the data. First of all, if you look at the images in the center here, which are past staining of glycogen, you can see that when the cells are starved, on the left-hand side, there's relatively weak staining. But after the cells are fed with fresh new media, which does include insulin, you can see that after 4 hours, on the right side, the past staining is much stronger, or a deeper pink or purple color. And this indicated to us that there was a lot of intrinsic metabolism going on here. So thinking about gluconeogenesis, there were a lot of parameters that could affect the assay, including the most appropriate precursor substrate or substrates to feed the cells, and how long to stimulate the cells with hormones to activate or inhibit gluconeogenesis. And after all this assay development, what we discovered was that many of the physiologically relevant molecules mentioned earlier really worked for these cells to be used as substrates to convert into glucose. A side note, we even had a customer who was working with these cells and in this sort of system, and he described them as metabolic monsters. But anyway, in this data shown here, the titration of insulin on the left and the dose response of glucagon on the right, we found that lactate ended up being the most important substrate for a robust gluconeogenesis assay, and treatment times of about 6 hours with either insulin or glucagon really gave the best results. Okay, this physiological readout of glucose production can be complemented by dissecting the cell signaling events involved following stimulation with insulin or glucagon. The insulin signaling pathway, shown here in the schematic on the left, is well characterized, and there are many biochemical tools available to quantify activation of the pathway, primarily by detection of phosphoproteins following the cascade of kinase events. Here we used assay kits from SysBio to look at increased phospho-AKT at both sites, threonine 308 and serine 473, after treatment with insulin. And we also observed a decrease in the assay signal when the pathway was blocked with the inhibitor PI-103. Additionally, in these bottom two graphs, the downstream phosphorylation events on mTOR and GSK3-beta were also responsive to the increasing concentrations of insulin. And these data together are a simple demonstration of how dissecting the phosphorylation state of these proteins can be used to understand mechanism, if necessary. And these data indicate that iCell hepatocytes have an intact insulin signaling pathway. Now for glucagon. The receptor is a GPCR and not a receptor tyrosine kinase, or RTK, as is the case for the insulin receptor. Stimulation of the glucagon receptor results in activation of adenylate cyclase and increased levels of cyclic AMP inside the cell. Glucagon receptors are mainly expressed in the liver, and since we saw earlier the functional response to glucagon in the gluconeogenesis assay, we sought to measure the second messenger cyclic AMP with the HTRF assay. And this is a simple and sensitive system, but you really have to think about the data a little bit backwards here, meaning that there is high fret in the absence of any cyclic AMP, with untreated cells or at low concentrations of glucagon. But when the cells are stimulated with an agonist, that is, with glucagon or these related ligands, and the levels of cyclic AMP increase in a dose-dependent manner, this breaks up the fret complex, and we observe a decrease in the signal. So a loss of signal in the assay indicates an increase in cyclic AMP production. And it's good to know that glucagon signals to cyclic AMP in these cells because it is known that the rate of gluconeogenesis is regulated through this second messenger pathway. And increased levels of cyclic AMP also promote glycogen breakdown. So these two influences on the production of glucose really could be studied in iPSC-derived hepatocytes. And remember, they could be studied in a normal individual or from a donor with some degree of metabolic disease. So I want to spend the last couple of slides tying this all together, going from really specific to opening it up to big picture. And this first example is that we have also measured insulin-dependent effects on gluconeogenesis in 3D spheroids of human iPSC-derived hepatocytes. Now, listening to that, I think that's actually pretty cool. And what this means is instead of using hepatocytes in 96-well format with about 30,000 cells per well, we can miniaturize the assay to use about only 2,000 cells in a 3D spheroid. And this takes advantage of the benefits of moving hepatocytes from 2D to 3D, as discussed earlier, and couple this with a very sensitive luminescent glucose detection assay. And then we can reproduce the gluconeogenesis result that was featured a few slides back. So this is something we are exploring further, but CDI has all the protocols in place if you are interested in running these sorts of assays. Okay, I would like to summarize by saying that even at this current state, human iPSC-derived hepatocytes definitely have a place in the game when it comes to the available cell models for studying some of the many different functions of the liver. And the chances are pretty high that CDI, in particular, has a great chance to drive this field forward given the high-quality human cells that we make, the time we spend optimizing conditions such as media for different differentiation and cell culture protocols, and the applications team that we have at CDI, which is dedicated to improving the functional performance of all of our cell types in any way possible. And now this connection to Fujifilm that is bringing some of the technology that was developed years ago for making color-negative film and applying it to biomedical research in the form of materials and scaffolds for 3D cell culture. And altogether, this really lays the foundation for a company to be able to provide the tools to build a complex liver model, as I suggested earlier in the webinar. So iPSC technology enables the generation of all the complementary and likely required cell types, such as macrophages, hepatoblasts, mesenchymal stem cells, and endothelial cells. And when placed into the right device or in the proper system, it is possible that amazing things will happen. So I will end there. Thank you very much for your attention. And I would like to thank the people we have collaborated with to generate many of these data. And we have worked with several different groups using iCell hepatocytes along the way, and their company logos were placed on the slides where appropriate. And I'd like to thank in advance Siobhan Mullaney from SBP. We are pleased to be presenting alongside her and are very much looking forward to her part of the webinar, because everyone knows that the second half is always better than the first half. So thanks again for your time, and goodbye.
Thanks, Cody. That was a great presentation to get us started. I think our audience now has a greater appreciation of the usefulness and versatility of iPSC-derived hepatocytes. Before we move on to Siobhan's presentation, I want to remind everyone to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. All right, with that, Siobhan, the floor is yours.
Thanks for joining today. My name is Siobhan Mullaney, and I'm going to talk about our drug discovery platform in human iPSC-derived hepatocytes as a model of liver disease. Our area of interest is in non-alcoholic fatty liver disease, NAFLD. This is certainly an area that has gotten a lot of attention in the pharmaceutical industry. It's a growing epidemic globally. As much as 30% of the U.S. population has non-alcoholic fatty liver disease, and obesity, type 2 diabetes, and hyperlipidemia are all associated with NAFLD. And so this patient population has a higher incidence of NAFLD. And about a third of that population is progressing to the more severe progressive form, which is non-alcoholic steatohepatitis, or NASH. And this can lead to liver-related morbidity and mortality. So there's been some great advancements in the clinic. However, the precise mechanisms leading to NASH remain poorly understood. And so we're interested in building a cell-based model in human iPSC-derived hepatocytes that is characteristic between the steatosis stage, which is a benign prognosis, and this NASH stage, sort of a pre-NASH model of this disease for drug discovery. The development of our cell phenotype in the iPSC hepatocytes involves a multi-pronged approach delivering insults to the liver that will deregulate de novo lipogenesis, and this is the pathway that's taking newly synthesized fatty acids and converting them into triglycerides, phospholipid, for storage and energy utilization. So we're exposing the cells to monosaturated fatty acid as well as saturated fatty acid, so indicative of a high-fat diet. And this is increasing the triglyceride content and free fatty acid pools to drive to steatosis. And saturated fatty acids are also creating stress on the endoplasmic reticulum, or the ER. So the ER is responsible for calcium homeostasis, lipid synthesis, storage, and export. And the stress of ER stress has been implicated in development of steatosis and progression of steatosis to NASH. So in addition to incubating the cells with free fatty acid mixture, we're also adding in thapsigargan, which directly works on the ER to block the cell's ability to pump in calcium. So what we've created is this vicious cycle in the liver, which is contributing to the de novo lipogenesis, lipid load, but also then impairing the ER's ability to restore lipogenesis and homeostasis. And so we get then driving the liver to fat accumulation, inflammation, and the ballooning of the liver, which is more of a phenotype for NASH. In terms of the cells themselves, the phenotype detection of the phenotype is a 2D high-content imaging platform. And we are showing the nuclei in blue Herx dye and the lipid content by using a Bo-Dippy green dye. So vehicle treatment of BSA-DMSO gives us our normal liver phenotype, and the mixture of oleic acid, palmitic acid, then you see an increase in that green color and lipid content, and this is more of a fatty liver steatosis phenotype. And then we're driving that even further with the addition of thapsigargan to have a steatotic phenotype with chronic ER stress, which is more of a pre-NASH phenotype with the multiple insults acting together. Our disease and dish strategy is twofold. So we've developed this robust 384-well high-content phenotypic platform using the human iPSC-derived hepatocytes, and we are looking at chemogenomic screening using an annotated compound set where we know what the compounds are acting on in terms of the gene family and target pathway. So by using this annotated library set on our phenotypic platform, we can understand polypharmacology and identify potential target pathways for NAFLD. On the other side, we've looked at transcriptomics and the gene expression profile of our treated cells versus our control cells, control treatment. And we have more than 300 differentially expressed genes. And by this approach, we can also look at potential target pathways. And so these are very powerful tools then to build in targets and pathways onto our phenotypic platform that is a human relevant disease model. We are using the iCell hepatocyte version 2. This is the first stage from the iCell hepatoblast, and we mature those, differentiate those for 1 week in culture, and this has just worked very well for us in terms of reproducibility and robust response in the cells, so we haven't deviated from this protocol. Our image and data analysis workflow incorporates the development of this platform with the 7-day culture of the cells, drug exposure, the free fatty acid treatment with thapsigargan, the 24-hour incubation. We fix the cells, stain them, and then do the image acquisition using an Operetta high-content imager in 20x objective in several fields per well. And we use a Columbus software system to do the image analysis to detect nuclei, whole cell, and spot detection which is proportional to the neutral lipid load, so we can quantify the amount of lipid with whole cell spot detection. This slide shows nicely the progression to this lipid loading in the cells. The top row from left to right is the oleic acid increasing, which you see very minimal green or lipid content. And the middle row is the addition of palmitic acid with oleic acid, and there's an increase, a nice increase in lipid content. And then certainly on the lower level, we now see when we add dapsogardin and increase palmitic acid, we see a significant increase in the lipid content. And the cells are still a nice monolayer and morphologically healthy, but still we see a nice lipid content. Quantification of that slide I just showed is shown here. We have a nice integrated spot signal fold change on the x-axis and an increase in the green bars for the free fatty acid. Dapsogardin alone, TG, in purple, shows a minimal increase in lipid spot signal. Then it's the combination of the dapsogardin and the free fatty acid mixture that we see this saturation and this tenfold difference in spot signal or lipid content, and this is really the highest we've seen for inducing lipid accumulation in hepatocytes without an overt cytotoxicity. And this is showing then as a screening platform, nice statistics between our positive control wells, which are these dapsogardin free fatty acid treated wells, versus the negative control wells, which is the BSA DMSO treated wells. So a nice dynamic range and Z primes in terms of screening at 0.5. And then there, in terms of an ATP content, we don't see an effect on cell viability, which is very important to note because we are really stressing the liver and the endoplasmic reticulum, which should be turning on apoptosis pathways. So we have this very delicate balance in stressing the cells to be moving toward a pre-NASH phenotype, but yet we don't have the apoptotic events or cytotoxicity occurring. And so to validate this platform, we used obeticholic acid, or IN747. This is in late trials for NASH. It's approved for bile duct injury, and it is a agonist of the nuclear receptor, FXR, which FXR deficient mice have been characterized by hepatocytosis. Agonists of FXR regulate glucose triglyceride metabolism. So this was a very nice clinical control to be able to have to show that it dose-dependently inhibited our phenotype, and with a potency range that's similar to reporter assays using IN747. Now that we have a very nice, robust screening platform with a strong phenotype, we've signed a material transfer agreement with AstraZeneca to use their annotated compound library set of 13,000 compounds. So these are compounds where it's known what target family they modulate or what target pathways they modulate. And so we'll be screening this library to understand from the hits and the non-hits together what we are modulating in our phenotypic screen. And using the iPSC hepatocytes from cellular dynamics. So using the normal iPSC hepatocytes has been a great tool and platform to develop the screening platform that's robust and reproducible and versatile. But the ultimate goal is to use patient-specific samples, and CDI has more recently gained access to several lines from the SERM bank that include lines with mutation directly connected to NASH, and they're currently developing those into iPSC-derived hepatocytes. So we really look forward to adding these cell lines to our drug discovery cascade. So post-screen, we can look at how our compounds would affect actual patient-specific iPSC-derived hepatocytes. So I've taken you through our one approach is to build this platform and use it as a screening drug discovery platform. And the other approach was transcriptomics and to investigate the differentially expressed genes in our treated cells. So we've done this RNA-seq profiling, and we've currently going through the expression data. This shows a volcanic plot here with iPathway guide, and we see about 323 differentially expressed genes, and so this shows in the red dots those that are either up or down regulated and that have significant fold change or p-values. And then we've since looked at more of the ER stress pathways and done gene clustering for individual genes. So it was good to know that of the differentially expressed genes, we had 11% of those, or the yellow pie piece shown here, that are directly related to the ER stress pathways. And so that really validates that we are turning on these pathways within the ER stress that should be linked to a liver steatosis pre-NASH phenotype. So honing in on the ER stress pathways, this shows there are three transmembrane protein pathways as shown here, and we are affecting, in terms of p-value in the dark blue, several genes within each of these different transmembrane proteins that are part of the unfolded protein response that helps the ER cope with overload. And we're seeing some significant fold change in the dark red color for many of these genes. So we're affecting really all three of the pathways within the unfolded protein pathway of the ER. So to take that even farther and break these out as each one of these pathways, that's shown here in this slide. So for example, in the PERK pathway, we have the red arrows showing those genes that are upregulated in our system of our treated cells with free fatty acid dapsogardin in comparison to our BSA-treated cells. So we see, for example, an upload of CHOP, which turns on apoptosis. We're also affecting the IRE1 pathway and XBOX protein 1. And so these pathways activate in the nucleus the transcription of a set of factors that allow restoration of the ER homeostasis. So if we're affecting really the ability of the ER to restore homeostasis, and that contributes to this dysregulation of de novo lipogenesis. The key points here, I've talked about our robust high-content phenotypic pre-NASH platform. Dapsogardin, the ER stressor, in combination with the free fatty acid, worsens the steatotic phenotype and is more, we believe, a pre-NASH phenotype. We have very good statistics for screening, as E' is 0.5 between our control and treated wells. And the clinical compound in 747 shows a nice dose-dependent inhibition of our phenotype, and this compound does play a role in controlling hepatosteatosis. On the transcriptome analysis side, there were 323 differentially expressed genes, and many of these were ER stress markers in the treated cells, and also targeting specifically the unfolded protein response pathway. All of this work was really performed by Maddalena Parafati. She's a research fellow from University of Qatar in Italy, very talented scientist, and has done amazing work on this project. We hope to publish her work soon. She's been supported internally by a diversified translational laboratory initiative, which has involved multiple investigators, including myself from translational biology, Ferdinand Razinazad, Sepeda Corazon-Zea, in metabolic disease area. And it's also supported in part by NIH R01 grant. Ada Koo, who was a previous researcher in my lab, originally developed the oleic acid lipid accumulation model and high content model for which all of this was really built out of, and she's gone on to pursue her PhD and work at Hong Kong University of Science and Technology. And so thanks to the Conrad Prebys Center for Chemical Genomics, the Genomic Core and Bioinformatics Core at Sanford Burnham Prebys, Cellular Dynamics, and AstraZeneca Open Innovation Program. And thank you for listening.
That was great. Thanks, Siobhan. You provided the audience with some great insight into the importance of liver disease research and how the iPSC-derived hepatocytes facilitated your work. Before we start the Q&A session, I want to let everyone know this is their final chance to submit your questions for our speakers. It looks like we've got some really great questions already, but I'm encouraging the audience to keep them coming. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, since we already have some really great questions, let's begin the Q&A so that we can get to as many of the questions as possible. Bear with us for a few moments while the presenters collect their thoughts and we get everything in order for the final part of the webinar.
All right, everyone, so let's start off with the first question. Kobe, the first question is for you. One of our audience members had said, I'm thinking about your slide that shows various stages of your iPSC differentiation. Approximately how long does it take those different hepatocyte products?
All right. Hey, hi there, Jeff. Thanks for the question. You know, each step basically takes about 10 days, really. So from hepatocyte to, from iPSC to hepatoblast takes 10 days. From hepatoblast to hepatocyte 2.0, as we call it, takes 10 days. And then working with hepatocyte 2.0s to get to this 1.0 stage takes about 7 to 10 days. So it's really about 30 days in total to get a hepatocyte that we can suggest to start working with.
All right, thank you, Kobe. Siobhan, question for you. How are you assessing cytotoxicity and apoptosis in cells after exposure to dapsogardin and palmitic acid?
Yeah, thanks for the question. It's very important. I did mention a little bit on the morphology side in the high content platform that we're looking at brightness and shape and size of nuclei. But in addition to that, because dapsogardin does turn on apoptosis and palmitic acid can cause lipotoxicity, that we further looked at live cell imaging using a caspase 3,7 dye for apoptosis. And we also looked at reactive oxygen species through live cell imaging as well. And so the morphology of the nuclei, as well as the ROS and caspase 3,7 activation, we didn't see in all of our treatments any significant effect in those assays. So we're pretty confident then that at least at the stage that we're testing these and treating these cells, that we're in a more cytoprotective mode, and we have not yet tapped into the cell death pathways.
Great, thanks, Siobhan. Kobe, next question for you, more of an important logistical question. One of our audience members would like to know how many cells of the iCell hepatocyte?
Yeah, so how many cells are in the vial of hepatocytes? So there are about 8 or 9 million cells in a vial. These are terminally differentiated. They are not proliferating to a degree that where you can keep culturing them and use them over and over. So one vial is enough for at least one 96-well plate in 2D format. And as I mentioned during the webinar, of course, you can get many more plates if you move to 3D cell culture. So 8 to 9 million per vial.
Great, thanks, Kobe. Siobhan, another question for you. Is lipid secretion modified in the treated cells?
Yes, great question, because this kind of starts to hit on potential mechanism because in our phenotypic platform we could be affecting uptake or synthesis, export, oxidation, a number of things. And so we have started to look into lipidomics and lipid biomarkers. I can say that in looking at the free fatty acid secretion in the media that we do see similar, very equivalent level between the treated cells and the untreated cells in the amount of free fatty acids secreted. So because we're adding in all this exogenous lipid in our treated cells, then that really suggests that we are actively uptaking the lipid. We're also seem to be impairing export. We have looked at levels of ApoB100, for example, which is an important lipoprotein that's part of the very low density lipoprotein vesicles and induced with ER stress. So it does look like we're actively uptaking and impairing export.
All righty, thank you, Siobhan. Kobe, another question for you. One of our audience members writes, one issue with isolated primary hepatocytes from animals is the rapid dedifferentiation after dissociation. What is the timeline of dedifferentiation for these iPS-derived hepatocytes?
Yeah, another good question, and that really goes to some of the limitations that I mentioned on one of the early slides about using primaries. Dedifferentiation means that the hepatocytes revert back to a more fibroblast-like phenotype or cell morphology. This happens within, a very short period of time after thaw with primaries, and this is, this common problem. They ultimately lose their hepatocyte morphology and their function. iPSC-derived hepatocytes, on the other hand, really can maintain their hepatocyte morphology for 3 to 4 weeks post-thaw. And, again, this isn't something that's a real quantitative measurement. This is how they look. But we've, kept our cells in culture and not seen any dedifferentiation up to nearly 4 weeks.
Siobhan, a question for you. One of our audience members asked for a little bit further explanation. They didn't understand what you were doing with the AstraZeneca compounds.
Right, yeah, thanks for that, because this, I think, is an important strategy, so happy to explain it. But AstraZeneca has a 13,000 compound set that's available to academic labs through an application process, and they know they have a lot of information about these compounds in terms of if they are kinase inhibitors or specific selectivity profiles. They have all of that collected into this set. So what's nice to use it on a phenotypic platform is that we can look at the compounds that decrease the lipid load as well as increase, so those would be anti-hits, as well as those that do nothing, and do bioinformatics based on the information for these compounds to really start to tease out what actual pathways or specific targets we might be hitting in the phenotypic platform.
All right, thanks, Siobhan. We have a couple more questions for you. One of our audience members asked, have you guys had a chance to test the assay conditions for the induced NAFLD or NASH model in other hepatocyte-like cells? For example, do you think this would work if you simply use the HepG2 cell line, or do you think INT-747 would display the same inhibitory pharmacology?
Yeah, this is a good question because obviously it's cheaper to use the HepG2 cells. They are a cancer cell line, so in that respect, we don't feel like they're as relevant as using the iPSC hepatocytes. In our hands, we don't see the reproducibility as well as we do in the iPSC hepatocytes. HepG2 cells require a lot of serum. The iPSC cells are serum-free. That's one of the big differences, so you'll see that in compound effect. Fortunately, we haven't tested INT-747 in the HepG2s, so I can't answer that question, but I think in terms of reproducibility and relevance, there's some significant advantages to using the IC hepatocytes.
All right, thanks, Siobhan. Another question for you. One of our audience members asked, are you differentiating the hepatocytes from the CRM lines, or is CDI making them for you?
So CDI is making those, they have access to the serum bank and are making those specific cells that harbor a mutation that's associated with NASH. They're actively making those into the iPSC hepatocytes. And so those will be, as I understand, available then to customers to use as part of their drug discovery platform.
All righty. Question for you, Siobhan. One of our audience members would like to know, how long does it take to induce the NASH phenotype?
Right. So we do a 24-hour incubation of the Dapsgargan or drug treatment, and it's 24 hours later that we do the staining and visualization of the cells.
All right. Another question, I believe this one is also for you, Siobhan. Can you clarify if you used iCell 1.0 hepatocytes in these applications and how their functions vary?
Yeah, we originally started with the iCell 1.0 hepatocytes and developed the original platform a couple of years ago with the lipid uptake, and then more recently, just because it was more available to use the 2.0, we reestablished the assay in that one. So really, I don't think we see much difference because we've been able to use both of them. They both uptake the lipid. It's just once we started using version 2.0, which has a longer differentiation time in culture, and got that protocol down, we've just stayed with that protocol. But we started with the 1.0, and we still had nice data. So in our hands, we don't really see a difference.
Hey, that's great. I'm just going to add something. Yep, I'll just add to Siobhan.
Please, yeah, go ahead, Kobe.
Perfect. So, yeah, I mean, the 2.0 product was developed just to really help, like I said, industrialize and scale up this differentiation process, and doing a little bit of extra culture time ahead of running the assay is really the main difference. So all the data that was presented in the first half with the glucose regulation was using the 2.0 version of the cells as well. And Siobhan did a great job answering that she had actually run the initial part of this assay development in both cell types and got very similar results. So that's good.
Alrighty, thank you guys. And we have one last question. One of the audience members would like to know, they were interested in using the technology to do hepatotoxicity studies, and they want to know if your methods were approved by the FDA.
So I can answer here. There are a lot of governmental agencies and regulatory parties that are dictate what to do in toxicity. So CIPA for cardiotox and HESI for neurotox, and we haven't. The iPS-derived hepatocytes haven't been in the discussion yet for hepatotoxicity, but we have lots of examples of how these are being used in hepatotox. There are several other publications and webinars and other things to help showcase how alternative models to primary human hepatocytes are used for hepatotoxicity. So we can help direct you to these resources offline. But great question, thanks.
All right, great. Thanks, Kobe. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site at www.genengnews.com for up to a year. So if you missed any parts of it, you can watch it again or watch it over, or you can feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Siobhan and Kobe again for their informative presentations, and I'd like to thank the audience for their attention and very thoughtful questions. And a very special thanks to CDI Technologies for sponsoring this webinar. So hopefully we'll see you again in the near future for another Gen webinar. Goodbye for now.